Thanks very much. Can you guys hear me okay back there? Everything okay, dialed in? Well, thanks. It's a pleasure to be here. I'm going to tell you this story on Arbidol that started over 10 years ago in my lab when I had a visiting virologist from Moscow who wanted to learn hepatitis C virus infection and replication assays. And he brought this drug, Arbidol, that's used clinically in Russia with him. So I'd like to first start by disclosing that I have no competing interests for this talk. And by way of overview, I'm going to tell you about Arbidol, its brief history as an anti-influenza virus drug. And then the bulk of my talk will be focused on the inhibition of other viruses by this drug, including hepatitis C, Ebola, Zika, and recent data that are in revision right now for Lassa virus. And I hope to convince you in my talk that this compound inhibits these diverse viruses by at least two mechanisms. And the principal mechanism, we think, is that it targets viral fusion glycoproteins to inhibit the last step of the virus entry process that occurs in with endosomal membranes. And the secondary mechanism is that we think Arbidol engages membranes, and that may suppress replication events that are dependent on viral proteins associating with intracellular membranes. So Arbidol, the structure is shown up here on the top right, is an indole-based compound. Here's its PubChem ID number. It's also known in some spheres as umifenovir. And it was developed in the late 80s by the Russian Research Chemical Institute as an anti-influenza drug. And since the early 90s, it's been available as an over-the-counter, orally administered drug in Russia. Hundreds of millions of doses have been given. They give it from kids to adults to the elderly to pregnant women. It's also popular in China. And this is what a current packaging of Arbidol looks like. These references here on the right represent some recent publications in Western peer-reviewed journals. Many of the publications on the original description of Arbidol are in Russia, so it's difficult, unless you have translation, to understand what's going on. They inhibit influenza virus, which is an enveloped virus with single-digit to low double-digit micromolar concentrations and ranges of doses in milligrams per kilogram per day in rodent models of influenza protection. A recent publication by Irina Leneva from Moscow and her colleagues compared the in vitro antiviral efficacy of Arbidol, also known as umifenovir, as I told you, with Tamiflu or also tamivir carboxylate. This study, they examined 36 seasonal isolates, and some of the Russian isolates from the 2010-2011 season are listed below. And you can see that Tamiflu inhibits most of these viruses with single-digit nanomolar potency, and Arbidol has low micromolar potency against these viruses. One thing I wanted to point out is that resistance to Tamiflu is known, and so this isolate from Australia, you can see that the IC50 shifts from 1 nanomolar to over 300 nanomolar. And in this case, Arbidol is able to suppress both the wild type and the Tamiflu-resistant virus. So this suggests that Arbidol can block influenza infection in cell culture. And Irina's study also looked at a lethal mouse model of flu infection where 100% of the mice died within 7 days. And so if you give Tamiflu at 10 milligrams per kilogram per day, you can get complete protection of the mice, reducing the dose to 2.5 mg per kg per day. Protects about 40% of mice. And Arbidol at 60 mg per kg protected 50% of mice. They also, in this study, again, this was focusing on the 2010-2011 flu season. Over 1,400 people were hospitalized in their study for suspected influenza illness. Over 400 of these patients were confirmed to have influenza infection. So they did a sort of a small clinical study where they gave people Arbidol, Tamiflu, or some people didn't receive treatment. This was a retrospective analysis. And so you can see when they looked at various clinical features, such as when they started drug, it was imperative to start the drug as soon as possible within detection of symptoms. So in this case, Arbidol and Tamiflu were given within a day of symptom onset. And you can see that these other factors, like length of hospitalization stay, was decreased by 2 to 3 days for both Arbidol and Tamiflu treated patients compared to the patients who didn't receive any regimens. And the illness duration was reduced by 1 to 2 days, as was the fever duration. So these are some examples from quite a bit of literature showing that Arbidol has pretty good antiviral efficacy against influenza virus in cell culture, in animal models, and in people. So since that time, we and others have been contributing to this growing literature of a large variety of viruses that are inhibited by Arbidol. So the virus family is here on the left. So there's many enveloped viruses. These have a lipid envelope with a viral glycoprotein that mediates receptor interactions and post-receptor binding entry events. It also is a pretty good inhibitor of picornaviruses. So polio and Coxsackie B viruses are inhibited. Again, they're sort of single-digit micromolar to low double-digit micromolar concentrations. Some of these viruses have been tested for antiviral efficacy in vivo. So hantavirus is inhibited by Arbidol. It didn't have any effect. Arbidol was ineffective against Crimean Congo hemorrhagic fever virus in a recent report. Coxsackie B5 virus was inhibited at 50 mg per kg per day. The viruses and studies highlighted in red are the ones that my lab has contributed with a great group of collaborators, and that's what I'd like to tell you about. So I'm going to tell you about Arbidol inhibition of Hepatitis viruses, including hepatitis C virus, filoviruses like Ebola virus, flaviviruses like Zika, and Arenaviruses like ARB and Lassa virus. So you probably know much about hepatitis C virus. There's over 3 million people with known infections here in this country, with over 70 million infections estimated worldwide. In the US, there is estimated chronic hep C infection causes over 10,000 deaths per year. People who have chronic hepatitis C virus infection are at increased risk of developing severe liver disease, including cirrhosis and hepatocellular carcinoma. And as a result, in many developed countries, it's the most common reason for liver transplantation. But there's been a boon to the field in the last 3 or 4 years, and drugs that have been approved since 2015 can cure pretty much every genotype infection at any stage of liver disease. So this is a huge boon for curative therapies for hepatitis C virus. But back in 2006, Yuri Borskin came to my lab, but before I get to that, I want to tell you basically why hepatitis C virus is a problem. The major reason is that 85% of acute infections are not cleared and progress to chronic viremia, 70% of which develop a variable spectrum of chronic hepatitis or liver inflammation from mild to severe presentation. The majority of these patients with chronic hepatitis do okay, but about 20 to 40% progress to liver cirrhosis that can lead to death, or the cirrhosis can progress to hepatocellular carcinoma that can also lead to death. So of these 85% of people who get chronic viremia, about 10 to 15% end up dying from as a result of end-stage liver failure. So before these new drugs came out in 2015, back in 2006, Yuri Borskin came to my lab because the hepatitis C virus infectious culture system had come out the year before, and he wanted to learn some virology to enhance his virology skills and take back to Moscow. So he learned the culture system, and he said, "Steve, can I test this drug?" And I said, "Sure." And so this is one example of what he found. This is a Western blot showing expression of the hepatitis C virus core protein. Cells were infected with this cell culture virus called JFH1. But before infection, cells were treated with Arbidol for 2 days before infection, 1 day before infection. Cells were also treated with drug at the same time infection, or the drug addition was delayed until 24 or 48 hours post-infection. So it's a very simple time of addition experiment, but it can tell you a lot about what's going on. And you can immediately see that cells that are pre-treated with the drug have a really great suppression of virus. You can also see good suppression of virus when drug is added at the same time as infection, and some reduction when you delay the addition of the drug to 24 hours post-infection. But the bottom line is it seems like the earlier the cells see the drug in the context of virus, the better things are in terms of suppressing the virus. So that got us thinking about that this drug might be targeting virus entry. So to tell you about that, I want to remind you, if you haven't, it's been a while since you've taken virology, this is the classic way of how viruses with lipid envelopes get into cells. They bind to a receptor, there's an endocytosis event into an endosome, and then there can be a release of the viral nucleocapsid and genome by pH-independent and dependent mechanisms. The viruses that we're going to focus on today all require an acidic endosome for a fusion event. And if you think about it, the viral genome is within a lipid envelope that's in an endosome. So the only way, and the cytoplasm is over here. So the only way that the genome can get through the membrane of the virus, through the endosomal membrane, is that these two membranes have to fuse. So if we look at, if we zoom in on this area here, this is sort of the schematics of how fusion of virus membranes with endosomal membranes happens. So the virus membrane is in blue, and the endosomal membrane here is in pink. And embedded in the virus membrane is the envelope glycoprotein. And all the viruses that we're talking about today have an envelope glycoprotein that mediates this type of function. So the envelope glycoprotein on these viruses mediates receptor binding, and it has a receptor binding domain. And what happens is in the endosome, there's a protease cleavage event which causes a structural change in this viral glycoprotein, exposing this fusion peptide that it inserts itself into the endosome membrane. And then there's an additional large conformational change where this glycoprotein folds back on itself, bringing the two membranes in closer and closer proximity, ultimately opening up a pore through which the viral genome can then leave the virus, get out of the endosome and into the cytoplasm to begin replication. So to start studying these entry events in more detail, we and collaborators here at the University of Washington and abroad have been using pseudovirus systems. And put simply, a pseudovirus is where you use another virus as a backbone and you replace that virus glycoprotein with your viral glycoprotein of interest that mediates this fusion. And in many cases, the structural components are from retroviruses such as HIV or murine leukemia virus, or there's other viruses like rhabdoviruses like vesicular stomatitis viruses. To these viruses are added a reporter gene, such as green fluorescent protein, luciferase, or beta-lactamase. And this is shown schematically on the left here, where there's a packaging vector. In this case, this is an example of a retroviral pseudovirus where the GAG, POL, and envelope genes are provided. And here, there's also some other alterations in it such that these viruses don't produce progeny viruses, so they're replication defective. The reporter construct is provided as a separate plasmid, and the third plasmid provides your viral glycoprotein of interest. So the cool advantage to this is that you can take the viral glycoprotein from a biosafety level 4 spacesuit, real nasty virus, and you can study the entry of that virus using that single viral glycoprotein. So in the end, you can make pseudoviruses against influenza, that express two viral glycoproteins, the hemagglutinin and the neuraminidase. Hepatitis C virus has two envelope glycoproteins. Zika has a single envelope glycoprotein. And Ebola and Lassa viruses also have a single glycoprotein known as GP. So working in collaboration with Eve Pecheur in France, Eve did some really nice liposome fusion experiments with these hepatitis C virus pseudoparticles. So the schematic of the assay here is that Eve constructed liposomes that were labeled with this fluorescent dye. It's a lipid dye. And the liposomes were so heavily labeled that they quench their own fluorescence. And so when the membranes of the pseudovirus fuses with the quenched liposome, the dye gets diluted across the two membranes, and there's a recovery of fluorescence. So you can see that for these two genotype 1b Hep C envelope glycoprotein pseudoviruses, there was a nice recovery of fluorescence, and Arbidol could dose-dependently inhibit that fusion event. And this was not unique to genotype 1 of hepatitis C, but the Arbidol was able to inhibit the 6 major genotypes to equivalent degrees. So this means that Arbidol can inhibit hepatitis C virus fusion with membranes. Eve, with Julie Blazing, a PhD student in her lab, also did a really elegant study. They tracked live virus particles by spinning disc confocal microscopy. And they looked at the association of hepatitis C virus particles labeled with a red dye with cells that were transfected to express a clathrin molecule that was tagged with green fluorescent protein. And clathrin is one of these proteins that is part of the endosome during the initial entry events. So what Eve and Julie could show is that the virus comes in, and after about 30 seconds it starts to associate with clathrin, as you can see this sort of co-localization. That lasts for about 90 seconds. The virus then leaves the clathrin endosome and moves to a deeper place in the cell. And with markers of early and late endosomes, they were able to prove that the Hep C was moving to a late endosome. And you can track these virus particles because this is happening in real time. So you can see the virus particles moving all over the place and ends up in the endosome. However, in the presence of Arbidol, hepatitis C virus seemed to remain associated with clathrin-coated structures and rarely made it to the early endosome. And you can see that the viral speed and mobility was greatly reduced in the presence of Arbidol. And when you quantify the association of the virus particle with clathrin, again, you can see that the virus associates in untreated cells for about 90 seconds with clathrin, but this time is really increased. The time of association of virus with clathrin pretty much stays the same throughout the experiment. So these data indicate that Arbidol can impair trafficking of hepatitis C virus in cells. So the story got a little more complicated because as I showed you that Uri first tested it against the infectious culture system for Hep C, but we decided to test it against a hepatitis C virus replication system. And so these cells called FL-NEO are a human liver cell line that has a genome-length hepatitis C virus replicon in it. And what a replicon is, it's simply a synthetic RNA that has all the enzymatic machinery of the virus, and it replicates viral RNA and protein continuously under a selectable marker. So you can see that Arbidol treatment, every other day Arbidol treatment over a course of weeks caused progressive decreases in hepatitis C virus non-structural and structural proteins. And when they measured, when Uri measured HCV RNA, you could see that the viral RNA eventually disappeared by 9 weeks of treatment. So in fact, Uri was able to cure this cell of the hepatitis C virus replicon. So these data indicate not only that Arbidol might be affecting virus entry, but it might also be affecting virus replication. How we think this is happening is described on this slide, and this is also a result of many collaboration with Eve's lab. We know that it can inhibit trafficking in cells. We also think there's good evidence now that it can inhibit this fusion process that I told you about. And we also have demonstrated that it can inhibit HCV replication. More specifically, where we think this is happening is on these intracellular membranes through which the viral replication complexes assemble. And I'll show you some data later that Arbidol can engage proteins on membranes. So, hepatitis C virus was going away, and I knew this years ago, so we started, we had Arbidol tested against a battery of viruses through the NIAID in vitro antiviral testing program. And during the 2014 to 2016 Ebola outbreak, we became interested in Ebola and Arbidol. So before I tell you the data, I want to tell you about Ebola virus disease. Or EVD. The average case fatality rate from Ebola is 50%, and it varies depending on the outbreak and the location, from 25 to 90%. The symptoms arise after an incubation period of 2 to 21 days. It starts as a classic sort of inflammatory antiviral response, onset of fever, muscle pain, headache, sore throat, that can then progress to some really nasty symptoms, including symptoms of impaired kidney and liver function. And in the late stages, there's internal and external bleeding, and people can just bleed out. So this is one of these classic hemorrhagic fever viruses that are the topic of all these sort of outbreak novels. Transmission is due to direct contact with blood, secretions, or other body fluids from infected people, or materials from these contaminated materials from these people. More recent studies have indicated that the virus can hang out in several reservoirs that may allow virus persistence and seeding of new outbreaks. For example, the virus has been found in the semen of men who recover from Ebola virus disease, and actual sexual transmission of Ebola has been reported. And the virus has also been found in the eyes of people and animals who recover from Ebola virus disease. So this slide summarizes the last, 30 plus years, 40 years of Ebola virus outbreaks from 1976 up to the 2014 outbreak. In terms of the number of infections, you can see, as many of you know, the 2014-2016 outbreak was the largest ever on record with over 28,000 infections. This is primarily in Western Africa. And you can see that the number of deaths in this outbreak was the highest ever. And this wasn't unique to Central and Western Africa. Because of global air travel, there were several Ebola cases that ended up on European and US soil. And so this is, most virologists know this, but we should remember that viruses, they come and go, and like the Terminator, they come back, to the point that there's actually an ongoing outbreak right now in the Congo, that as of January 18, they're almost at over 600 cases of Ebola virus diseases with a case fatality ratio of 61%. So this is a nasty virus. It's a big outbreak going on in a war-torn country, so it's hard to get people in to support those who are infected. So during this, the 2014-2016 outbreak, I managed to convince Chad Meyer and Tom Geisbert at University of Texas Medical Branch in Galveston, where they have a biosafety level 4 laboratory, to test Arbidol against in vitro Ebola infection. So similar to what we'd learned, based on what we'd learned with hepatitis C virus, we came up with this experimental design where cells were treated for 24 hours before infection at the same time as infection, or we delayed the addition of drug for 1 hour or 24 hours post-infection. And you can see that pre-treatment the day before infection caused over 4 log drop in viral infectivity in terms of infectious virus that was coming out of these infected cultures. And if you added the virus at 1 hour at the same time as infection or 1 hour post-infection, there was a similar dose-dependent decline with some attenuation of antiviral activity at the higher doses. Really, delaying till 24 hours really didn't have a significant antiviral effect compared to these other two. I will note that there was dose-dependent inhibition, but it was less efficacious than when drug was added early. To look at virus entry in more detail, based on these results, Peter Hathman and Yoshi Kawaoke did an experiment with sort of like a pseudovirus, except they have a recombinant vesicular stomatitis virus that instead of having its own glycoprotein, they replace it with the Ebola virus glycoprotein. And we did the design of adding drugs to cells an hour before at the same time as infection, or delaying it till 1 hour post-infection. And you can see that all three treatment regimens, Arbidol suppressed the virus coming out of these infected cells, but the pretreatment was the most effective. And interestingly, as you delayed the addition of Arbidol, the effect became attenuated. So these data suggest, like the hepatitis C virus story, that we're blocking this later stage of the virus entry pathway. And this is just a little summary of how Ebola gets into cells through various receptors. It's endocytosed, not through standard endocytosis, but through macropinocytosis, because these virus particles are much larger. And once it also requires this acidic endosome for this fusion event that's mediated by the Ebola virus glycoprotein. So we think that's what's happening with Ebola. And so, like any good journalist, any good virologist will follow the latest trends. So the Ebola epidemic was replaced by the Zika virus outbreak that involved over 47 countries and tens of thousands of infections. And usually Zika infection, the symptoms are mild, such as fever, rash, joint pain, and conjunctivitis. And as we probably all know now, it's transmitted through the bite of the infected Aedes aegypti, a mosquito. Like Ebola virus, there's sexual transmission of Zika has been reported, and the virus is found in many, if not most, body fluids. The real problem with the American outbreak of Zika virus disease in the Americas, is that there was neurological damage in humans in the form of Guillain-Barré syndrome and in developing fetuses with the increases in microcephaly reported in Brazil and many other countries. So I wanted to tell you before we show you the Arbidol results and to remind you that Zika is a flavivirus. And the flaviviruses are grouped into mosquito-borne flaviviruses, insect-specific flaviviruses, and tick-borne flaviviruses. And over here is the Hepatitis virus, of which hepatitis C virus belongs. Hepatitis C virus has its own branch on the Zika family tree. So over here in more detail, you can see Zika is a member of this family of viruses that are spread by mosquitoes, including West Nile virus and dengue virus. And here's hepatitis C virus down here in the Hepatitis virus branch of this tree. So Zika came out. I brought a whole bunch of viruses into the lab and got approval from EH&S, of course, to do this. And we initially tested the original Uganda isolate where the virus was discovered in the Zika forest in Uganda. This strain is called MR766, and you can see that Arbidol causes dose-dependent inhibition of infection in these human hepatoma cells. And we did this in collaboration with Susan Fink and Connor Jackson here in Lab Medicine, and Lucia Vojtek, who's also here at the UW. We tested viro cells. Zika is a lytic virus, so you can see that if you infect cells and wait a couple days, most of the cells die. But when you infect cells in the presence of Arbidol, you can see that the virus-induced cytopathic effect is inhibited. And Chris Bassler and Sudip Kadka at Georgia State University helped us test it in another human cell line called A549. And you can see that Arbidol caused this dose-dependent inhibition of Zika infection, with pretreatment giving slightly improved IC50 over a 1 hour pretreatment. And so these are all established mammalian cell lines, and so we wanted to look at something that's a little bit more biologically relevant. And so with Lucia Vojtek and Peter Wang in her lab, Lucia and Peter, they study sexual transmission of viruses. So they have culture systems for primary human vaginal and cervical epithelial cells. And so you can see that that Zika robustly infected these human vaginal epithelial cells and cervical cervical endoderm and ectodermal cells, and that you could see that Arbidol suppressed the infection of these cells. And this is both the African lineage, the Uganda strain, as well as an Asian lineage isolate, which is the strain of virus that's causing all this microcephaly and all these other birth defects. And this wasn't just at the level of protein, but if you look at the amount of virus coming out of these cultures, you can see that Arbidol-treated cells cause log-fold reductions in infectivity. So we went back to our standard thing, which is a time of addition of experiment, and you can immediately see that 24 hours pre-treatment pretty much wipes out infection, and that there is, if you add virus or drug around the time of virus infection, there's also pretty good antiviral effects. But like what we've seen with Ebola and especially with Hep C, you can see some residual antiviral efficacy when you delay the addition of drug till 24 hours post-infection. So we think these are entry, inhibition of entry. This might be inhibition of replication. So to look at entry in more detail, as I mentioned, we collaborated and continuing to collaborate with Susan Fink and Connor Jackson, who've got a different type of pseudovirus system. Here, a West Nile virus replicon that expresses green fluorescent protein is packaged with the structural components of Zika from the Ugandan isolate. And when you do this, you deliver these viruses you infect cells with these viruses, and if you look 24 hours later, the cells, if they get infected, will express green fluorescent protein. And you can see this here by flow cytometry, that when you infect cells with the Zika pseudovirus, you get green fluorescent protein expression, and if you treat with an inhibitor of virus entry, you can reduce that fluorescence. So when we do this with Arbidol, there's also a dose-dependent inhibition of GFP-positive cells. Finally, but we're still getting to mechanism. I'm just giving you some hand-waving things about mechanisms. I wanted to tell you about new data with arenaviruses. And in our 2016 paper with Yves Picheur, the focus of the paper was on Ebola virus, because that was the virus du jour back in 2014, 2016. But we also got a hit from the NIAID screening program against Tacaribi, which is a New World arenavirus. And that kind of rang a bell for me, because a long time ago, before I moved here, I did my PhD on arenaviruses, on Pichinde arenavirus. So we started thinking about arenaviruses, and I went and reread my thesis and remembered that arenaviruses cause human disease, the most famous of which, if you're an immunologist, you know lymphocytic chorial meningitis virus, or LCMV, which was discovered many years ago. There are many viruses in South America, like Hunan virus, Machupo, Guanarido, Sabia, Chapare, and Lujo viruses that cause various, they're named after the location that they were discovered. So Hunan causes Argentine hemorrhagic fever. And this other one, which is probably the nastiest of all arenaviruses, is Lassa virus that was discovered in the Lassa Valley in Africa, and it causes Lassa fever, which is a similar hemorrhagic illness to what Ebola causes, and this can also be fatal. So arenaviruses are found all over the world. As I mentioned, Lassa is kind of mainly Central and West Africa. You can see many of these viruses are, arenaviruses were discovered in South America. So Machupo is here. Pichinde is from the Pichinde Valley in Colombia. You can also see Hunan virus is down here in Argentina. So in terms of the diseases that Lassa fever causes, this is what you can download from the WHO. Similar to any virus infection, you get fever, headache, sore throat, chest, muscle pain, and then it can progress to nausea, vomiting, diarrhea. And in severe cases, people can bleed out. You can bleed from the mouth, nose, vagina, or gastrointestinal tract. What I thought, I wanted to put a little bit of a plug in for my cat, Eudora, because it turns out that Lassa is spread by rats or feces and urine from infected rats. And so the way to prevent it is to wash your hands, keep your home clean, and keep a cat. So, and it's got this picture of a cat, but I decided to put in a picture of my real cat. and let you know that Eudora is, you can tell she's vigilantly on guard against Lassa in my house. All kidding aside, this is a nasty virus, and in collaboration with Chris Hulsberg and Judy White's lab at University of Virginia, Judy is an expert in filovirus and arenavirus entry, so she had some murine leukemia virus pseudoviruses decorated with Lassa or Ebola virus glycoprotein, and you can see that there was potent inhibition of these viruses, and in fact, Arbidol seems to be an even better inhibitor of Lassa than Ebola virus, and this is very similar to the IC50 that we showed for live virus. Liz Nelson in Judy's lab recently showed in these data that we're hoping is going to be accepted soon in Journal of Virology, that it's not just that the inhibition of Lassa is not just unique to Lassa, in that Hunin pseudoviruses, LCMV pseudoviruses were inhibited similar to Lassa, although it seemed to be a really good inhibitor of Hunin virus. And interestingly enough, just for comparison, Liz compared Lassa pseudovirus inhibition by Arbidol with flu pseudoparticles, and Arbidol was a considerably better inhibitor of Lassa than flu. And finally, with Jason Shoemaker, who's a research scientist at US AMRID, who also have a BSL-4 facility, Jason tested Arbidol against live Lassa infection, and again, we saw dose-dependent inhibition. I should say, we always do cytotoxicity controls, and so this is not because we're killing cells that we're also killing virus. So we do these experiments with great care in terms of drug-induced cytotoxicity. So now, with the few minutes that I have left, I want to give you some ideas about how I think this one drug can do so many things to so many divergent viruses. And the first thing is yet another study from Ev's lab showing that Arbidol, this was an NMR study showing that these protons 2, 3, and 4 can insert fairly deeply into the membrane, but it really interacts at the level of the phospholipid head. So here's the Arbidol molecule beside a phospholipid in the membranes. And this was a computational study that took into consideration the protonation form of Arbidol. And this group showed that the protonated form goes into membranes in parallel, where the non-protonated form doesn't really penetrate into membranes because it goes in perpendicularly. So this is of relevance when you think about an acidic endosome, where low pH Arbidol may be the protonated form of Arbidol may be the predominant form. Ev also showed that Arbidol can interact with aromatic amino acids. And so what she did here is she took a water-soluble tryptophan derivative called tryptophan octyl ester, and she inserted that into liposomes. And when you do that, tryptophan has a natural fluorescence shown in these bolded bars. And you could see that Arbidol dose-dependently inhibited this tryptophan fluorescence, meaning that it's somehow engaging tryptophan when it's in this membrane. And then what she also did, I apologize that this is cut off down here, but there's a sequence of a peptide down here that is the transmembrane helix of the hepatitis C virus NS4A protein. As I told you, many viral proteins insert into membranes as part of the replication complex. And so when she built liposomes with this transmembrane helix that has a tryptophan residue in it, again, there's an intrinsic fluorescence of this liposome protein complex, and Arbidol could dose-dependently inhibit that. So that means that this compound interacts with amino acids and proteins that are in membranes. And on the virus side, it's really hard to do, but in cell culture, you can select for drug-resistant mutations. And so this study, they selected for mutations against Arbidol-resistant mutations in chikungunya virus. And chikungunya virus is one of these, another mosquito-transmitted virus. So you can see the wild-type virus is pretty sensitive to Arbidol, but when they passage the infected cells for 17 passages over 4 months, they were able to isolate this drug-resistant mutant. And when they sequenced the virus, they found a mutation in the envelope glycoprotein, that mediates fusion. Arenaviruses also did a study where she selected Arbidol-resistant influenza viruses and found that all of the resistant mutations mapped to the hemagglutinin protein. Again, the hemagglutinin protein is the fusion protein. But the real icing on the cake is the piece of data that came out a couple years ago from Ian Wilson's lab at Scripps, where he and his colleagues solved the crystal structure of Arbidol bound to the fusion protein of influenza, the hemagglutinin protein. And so you can see here's the fusion protein in its pre-fusion state. It's actually a trimer, and 3 molecules bind to the trimer, so it's a 1-to-1 stoichiometry. And you can see some pretty pictures of where Arbidol is binding. And they went on to show, they did some really neat biochemistry experiments to show that when Arbidol binds to hemagglutinin, it prevents conformational changes associated with low pH. So I think this is the strongest data that where we think Arbidol is acting, it's binding to these viral glycoproteins and keeping it locked down in this prefusion state and preventing this large conformational change that is required to bring the two membranes together. However, as I told you, there's pretty convincing evidence that we can see some inhibition of virus replication when we add the drug post-infection, at post-entry steps, or at steps in the virus life cycle after virus entry. And because the virus can bind to membrane, the drug can bind to membranes, and the drug can bind to proteins in membranes, we think it might be inhibiting replication by interfering with these protein membrane interactions that are required for virus replication. And this is just a cartoon of what's known for many flaviviruses, including hepatitis C virus, Zika, and dengue, that there are these ultrastructural intracellular membrane rearrangements that are formed when the virus replication complex is set up on these intracellular membranes. So we think Arbidol might be inhibiting these. So in summary, Arbidol is a clinically used anti-influenza drug. It inhibits many viruses, both positive and negative stranded, and ambisense viruses like the Arenaviruses. Mostly inhibits enveloped viruses, but there are some non-enveloped viruses that are inhibited. It inhibits these globally relevant pathogens like hepatitis C, Ebola, Zika, and Arenaviruses. And we think it's mainly doing this by inhibiting viral glycoprotein fusion that brings endosomal membranes and viral membranes together. But we think because of its membrane interfering activities, it may impinge on virus replication. So this is a large collaborative effort without a formal source of funding, but we're working to change that. And I really want to thank Jessica Wagner in my lab. She's been on this Arbidol project that really started as a hobby, but we got more and more into it. More recent collaborations with Susan Fink and Connor Jackson here in laboratory medicine, I'm very grateful for. Lucia and Peter in Lucia's lab has been great to collaborate with. And we've had many-year collaboration with Eve Pecheur in France, and Arena here in Moscow. Well, they're in Moscow, but this is her picture here. A new collaboration with Judy White and her former grad student Chris Hulsberg, who went through the military to get her PhD and is now working at USAMRID, as well as Liz Nelson in her lab. Chad and Tom at UTMB Galveston in their spacesuits doing the Ebola work. And Jason Shoemaker was the guy who did the Lassa infection experiments in Connie Smalljohn's lab at USAMRID. And Chris Bassler and his team at Georgia State helped out for some of the Zika experiments. And I'd also want to thank Peter Hathman and Yoshi Kawaoke for some of the VSV Ebola glycoprotein studies. And a very long-term musical and scientific collaboration with Mark Harris at University of Leeds. We're also known, Steve Polak and Mark Harris, were also known as Stark Polaris at Hepatitis C virus international meetings. So if you're ever at a Hep C meeting, come see us play. I really want to give a shout out to my department, the Department of Laboratory Medicine, who has provided support at several times during my career to help me keep things going. So I'm extremely grateful for that. So I'll end it here and take any questions. Thank you.
Arbidol have an anti-HIV effect?
Yeah, the question was, does Arbidol have an anti-HIV effect? We tested it when Jan McClure was in the lab. It didn't really have any effect. And also Greg Melikian at Emory, he tested it several years ago, and we didn't see anything.
So I have a question about the it seemed like in some of your experiments you repeatedly gave Arbidol, even though the first application might have come after the virus was applied. When I looked at the data, I thought, well, it makes sense that it might not inhibit the first round of invasion and production of virus, but shouldn't it inhibit subsequent rounds of invasion? Did I misunderstand that, or is that not how it works?
No, well, no, that's a really good question. So the way, if we do so we always remove the virus inoculum. So whenever whether we add drug before or at the same time, when the virus goes on, it's on there, depending on the virus, an hour or a few hours, but we always take it off. That's just the way we do standard virology. And then we always add the drug back afterwards. So the principle behind that is you remove the inoculum, you remove the unabsorbed virus. So the drug is always present after infection. So when we add back fresh media after we remove the virus, the drug is there as well.
So I'm just curious about if this drug works well, what's the process for getting it approved for use in the Western world? It sounds like it's used extensively in Russia and China.
Yeah.
And does that change everything, or does that make it harder because no one wants to work on a drug that they can't patent and have exclusive rights to?
Definitely for Ebola and Lassa, I think a lot of drug companies don't want it because it's not a moneymaker to give drugs, in resource-limited countries. From a global health perspective, what I'd love to do is, it's used clinically, the PK profile is favorable, many things about it clinically are known. I'd love to, get into Congo and do some sort of, prophylactic trial, or, for the healthcare workers who go there to give Arbidol prophylactically. That takes money and someone with a global health vision. In terms of the intellectual property, that might also be an issue. It's been around for so long, so I don't know, I'm sure anybody could make it. I mean, it's easy to synthesize. I would love to enroll a clinician. I just cut out. Are we back? Yeah. So I think it's, I'm the humanitarian in me says we should be using this now. The challenge is how to do that, because it still takes money. Whether you want to help people or make money, it still takes money.
What a great story. Thank you, Steve. One of the striking things about Ebola is the infectivity of the skin virus and then potentially absorption. What do you think about an effect topically in preventing transmission? Do you think there's a potential role?
Oh, you mean for sort of the arthropod-borne viruses?
I was actually thinking for Ebola, which is apparently infectious on the skin and through the mucus, through skin secretion, infecting keratinocytes in the skin. Would a topical application prevent absorption? Because for healthcare workers and for family members, that's a huge problem in the field. And I wonder whether you think there'd be enough binding to the virus and or prevention of transmission if it were applied topically. It sounds like it's inexpensive. I'm thinking about it it's almost a little bit like a detergent effect.
Right. So there's a couple. So first of all, that's a great question. Hadn't actually thought about it sort of for topical. It's certainly a question that we could answer with experimentation. I mean, you might, for the sexual transmission of it, I don't know if it's if it would work well on mucous membranes or something like that. It might be irritant because it is sort of like it's a indole compound. And but in regards to, what was your second question?
Well, this was related to again the problem
a detergent,
and family members getting exposed to the skin, infected skin.
So, I don't know. So your detergent question makes me think about, is it virucidal? And we get that question a lot. And there's been a few studies in the literature that have tried to look at virucidal effects. In other words, is it basically just neutralizing, is it neutralizing the virus? The studies haven't been done properly. There's many controls missing. We just, for this paper that we're revising with Judy White, we just did an experiment with Lassa pseudoviruses and basically just treated virus at a dose that would inactivate and then diluted it so it's the dose of drug is not antiviral. And you can see it's only when the cells are incubated that there's an antiviral effect. So at least for Lassa, and one of Ev's papers has shown that it doesn't seem to be virucidal. So it's not just killing virus particles. But in terms of preventing transmission through skin, that would be interesting. We just have to get a system.
So I have a question about the influenza. Is it effective against H3 or any other influenza? Because then that would be why it'd be less effective as far as a universal drug.
Right. Yes, there is something in Ian Wilson's paper about how it engages H3 versus H1, or I'm not a flu expert. So there is some differences in the way it engages. I can't remember them right now, but I'd be happy to look it up and communicate that to you.
Thanks, Steve.
Right. Thanks very much.